Welcome everybody to this very interesting event. The time is now 1:30 and today we are going to deep dive into Cobitolimod's mechanism of action. And with me today I have the CEO of InDex Pharmaceuticals, Peter Sahuni, and joined by Charlotte Admyr, the senior scientist within Cobitolimod's field. And also welcome to the audience today. And as always, you are, of course, welcome to post questions. And those questions you post on your right lower side of the screen. And I will forward the questions to Peter and Charlotte. With those words, I would like to hand over the presentation to Peter Sahuni. Welcome, Peter and Charlotte.
Thank you, Klaus. So my name is Peter Sahuni. I'm the CEO of InDex. And today I'm joined by Dr. Charlotte Admyr, who is an immunologist and a senior scientist at InDex, and has been with the company for a long time following the development of Cobitolimod. And she will provide a deep dive then into Cobitolimod's mechanism of action. So we will get quite technical today. First, we have our standard disclaimer regarding forward-looking statements. But I thought I would give a brief introduction to the company before we let Charlotte dive into the mechanism of action. So InDex is a Swedish biotech company that is developing a drug to treat ulcerative colitis to be able to help patients like Felicia here to live a normal life. And we do that primarily by developing our lead drug candidate Cobitolimod for ulcerative colitis, where we are now in phase 3 development. And phase 3 is the last stage of development before you can apply for marketing approval and commercialize the drug. Apart from Cobitolimod, we have a broad portfolio of preclinical early stage assets from our DIMS platform that actually share the mechanism of action with Cobitolimod. So it's applicable to all of them. And DIMS stands for DNA-based immunomodulatory sequences. And these have potential in many different immunological diseases. InDex is listed, or the share of InDex is listed on the Nasdaq First North Growth Market in Stockholm since 5 years. And currently, I say currently because of the volatility on the stock markets today and recently, our market cap is 700 million Swedish crowns. Last year, we raised a bit more than 500 million Swedish crowns in an oversubscribed rights issue to fund phase 3 development of Cobitolimod. And at the same time, we strengthened the shareholder base. So today, our main shareholders are Link. That's the investment vehicle of Bengt Jolander, specialist life science investor in Sweden. We have the Swiss life science specialist fund HBM. We have the Fourth National Pension Fund of Sweden, and we have Handelsbanken funds. Together, they hold about 40% of the shares and capital in the company. But in addition to those, we have more than 5,000 shareholders and a lot of retail shareholders. So I thought I would give a brief explanation of what ulcerative colitis is, so you understand the problem we're addressing. Ulcerative colitis is caused by chronic inflammation of the colonic mucosa, so the colon, the large intestine, that leads to ulcers, and then the colon doesn't work. It's a lifelong disease that you have. There is no cure today. And it comes in, did that mean something, Klaus? There was a
No.
No. It has active periods and inactive periods. And when you're in an active period or a flare, the patients suffer from the typical symptoms of very frequent blood and mucus mixed diarrhea. And this has a significant negative impact on the quality of life for these patients. And we have a testimony here from Felicia how it is to live with the disease. There are a number of therapies available to treat ulcerative colitis and moderate to severe ulcerative colitis, which are the more difficult cases where we are active. But they have limited efficacy, so they help a fairly small share of patients, and they can cause severe side effects. So there are many patients that do not respond or suffer these side effects, so there's a high unmet medical need for new, efficacious, and safe drugs with new mechanisms of action. And this is where we come in with Cobitolimod then at InDex. So we're developing Cobitolimod as a potential new drug for moderate to severe ulcerative colitis. And as I said, we're now in phase 3. Our most recent study was a phase 2b study where we met the primary endpoint with an excellent safety profile, and this is now what we base the phase 3 study on. In addition to that phase 2b study, we've completed 4 previous clinical studies that support the efficacy and safety that we saw in phase 2b and that we now will prove in the phase 3 study. So what Cobitolimod has to offer is a competitive efficacy, at least as good as the currently approved drugs, but with a much superior safety profile. And that safety profile is probably to an extent explained by the way we give this drug. It's a local treatment. We give it by an enema, and you have a picture of the enema bottle there, that we give 50 milliliters of solution straight to the inflamed colon, but not very often, so just once every 3 weeks. So it's quite a convenient treatment. And as Charlotte will speak a lot about, we have a novel mechanism of action. And this good safety profile and the novel mechanism of action makes this a very good candidate for combination therapy in the future. So we see this primarily as a monotherapy, but there is also more and more talk in the field about combining several advanced drugs, and then Cobitolimod would be a natural choice, we think. So all in all, we see there is a significant commercial potential here with a blockbuster potential, meaning that we estimate Cobitolimod can sell for more than $1 billion per year, given this outstanding combination of efficacy and safety. Then, since we had our year-end report released yesterday, I thought I would give some comments to that as well. The most important thing that happened during the last quarter was that we included the first patient in the phase 3 study that we call Conclude, which was a very important milestone for the asset and for the company. In total, the study will comprise about 440 patients from a couple of hundred clinics in more than 30 countries. So it's quite a project, and in this startup phase where we're now, our focus is really on getting the trial approved in each of these countries and getting the clinics in the approved countries activated so they can actually start recruiting patients. Then gradually the focus will shift towards the actual patient recruitment and getting these patients in as quickly as possible. We estimate that it will take 18 to 24 months to complete the study. We have seen the pandemic has affected the startup of clinical trials, but now we see that that is easing up, and a lot of clinics are really eager to get going. We also have now this issue of Russia invading Ukraine. And for us, we are working with a global CRO called Parexel that actually won an award recently. They were ranked the best CRO to work with by clinics worldwide. And they have quite significant operations in both Russia and Ukraine, and they are very active in this issue now. They have task forces, and they've taken measures to to adapt. So they built up extra stockpiles of material, and they have a lot of contingency planning done already. So we really hope that we can do this study in these countries. They are typically high recruiting countries, but as I said, this study will be in more than 30 countries, so they're not determinant for this. But of course, it would be great if we can continue there, and otherwise we are looking at backup countries that we can use in collaboration with Parexel, who is in almost every country in the world. So, but of course, a big tragedy now in Ukraine. We also enrolled the first patient in a smaller pharmacokinetic study with Cobitolimod end of last year. That's a much smaller study, about 6 patients, that will run in parallel with the big phase 3 study. And this PK study, the aim for us is to confirm that the systemic uptake of Cobitolimod is limited. We've seen that in previous studies, but now confirm that with the 500 milligram dose in an inflamed colon. And that would be that is a great competitive advantage, because the competitor drugs are all given systemically, meaning they affect the whole body and they can cause adverse events outside of the inflamed colon, which is the actual problem the patient has. So that will be interesting to follow. Then we got some additional patents granted for Cobitolimod during the quarter. And at the end of the year, we had 428 million Swedish crowns of cash following that rights issue we did. So we're well capitalized and financed until the readout of this first phase 3 study that we initiated now. We also last week invited for a Capital Markets Day on March 14. So I really encourage you to join that. And now we can actually have physical meetings. So you're welcome to Stockholm to participate in person. But it will also be webcasted. So you can't come, you can follow it online.
Thank you, Peter.
Thank you. Now I thought I would hand over to Lotta then.
Thank you very much, Peter. So I will then now give an overview of Cobitolimod's mechanism of action. So Cobitolimod is a first-in-class toll-like receptor 9 agonist, and toll-like receptor 9 is the immune system receptor for recognizing microbial DNA, so DNA from viruses and bacteria. And Cobitolimod is an oligonucleotide, so it's a piece of single-stranded DNA, which by mimicking a DNA from a good bacteria can activate this receptor. And this will lead to modulation of the immune system, which I will show you later on in the presentation. And that will lead to a local anti-inflammatory effect and healing of the colonic mucosa. Depending on the sequence of an oligonucleotide, different immunomodulatory effects can be induced through TLR9. And as Peter mentioned, we at InDex has designed over 100 oligos, which we call DIMS compounds, with different sequences. And in this way, we can modulate the immune system in different ways. But with Cobitolimod, we clearly see an anti-inflammatory effect, which I will show you on the later slides. So Cobitolimod has a novel and unique mechanism of action for treating ulcerative colitis. And there are several advantages with having this novel and unique mechanism of action. First of all, you do not have any competition for your mechanism of action. You are not another anti-TNF or another JAK inhibitor. You are unique. You can also address patients that have failed other mechanism of actions. And as Peter mentioned previously, you also have the potential to combine it with therapies with other mechanisms of action, especially as Cobitolimod has a very good safety profile. So we have investigated Cobitolimod's mechanism of action using different model systems for ulcerative colitis. We have both used animal models, We have also used cells from ulcerative colitis patients in vitro, so in the test tube, and also by using biopsies, so small pieces of the colonic mucosa that we have obtained from patients in our clinical study. And I will show you some of the data that we have obtained through this analysis. And as a start, the most common animal model to use for studying experimental colitis is a mouse model called the DSS model. And in this model, you induce colitis in the mice by giving them a substance called DSS in their drinking water. This DSS will then reach the colon and induce inflammation there, and they will also develop ulcers. So it mimics the clinical picture that you see in ulcerative colitis patients. And let's see if my pointer works here. I was going to point here.
It works a lot.
It works, great. So to measure disease activity in this animal model, you use a specific index called disease activity index. So the higher level of this index you have, the more severely ill are the mice. And here we have treated the mice with either placebo or Cobitolimod rectally to mimic the administration route that we use in patients. And they have given 2 doses, so at day 4 and day 8. And the black bar here are healthy mice, so they should not have any inflammation. And as you see, they do not have any disease activity index. We can see that the orange line here, which are the placebo-treated mice, they have increased levels of these disease activity index that are increasing over time. And what we can see is that mice that have instead been treated with Cobitolimod has a significant reduction in this disease activity index, meaning that they have improved their disease severity. If we at day 10 look in the colonic mucosa of these mice and see what immunomodulatory effects do we see, we can see that mice that has been treated with the DSS and been given placebo has increased level of inflammatory cells called Th17 cells. They are characterized by a specific marker called ROR gamma T. And these cells are known to be very much involved in driving the inflammation in ulcerative colitis. And what we can see that in mice that has been treated with Cobitolimod, we have a significant reduction of these cells. We also see a significant reduction compared to placebo in mice treated with Cobitolimod in IL-17 in the colonic mucosa. And IL-17 is a pro-inflammatory cytokine that is known to be produced by these cells. So here we can see both the reduction of these inflammatory cells and also inflammatory cytokines. At the same time, we also see an increase in T cells producing IL-10 after Cobitolimod treatment. And IL-10 is a known anti-inflammatory cytokine, so that is known to reduce the inflammation and dampen the immune response. So in this animal model, we see improvement of the disease severity. We see a reduction of pro-inflammatory cells and cytokine and an increase in anti-inflammatory cells and cytokines. So we have also studied the mechanism of action of Cobitolimod in vitro, so in the test tube. And here we have taken cells from ulcerative colitis patients. So we have isolated immune cells from the colonic mucosa of ulcerative colitis patients. Then we have stimulated them with a substance called LPS, and LPS then induces an inflammatory-like response, so that is to mimicking inflammation in the test tube. And then we can see that by adding LPS, we induce pro-inflammatory cytokines here in the orange bars. But if we add Cobitolimod to these LPS-stimulated cells, we instead see a significant reduction of pro-inflammatory cytokines. Again, we see a reduction of IL-17, which we also saw in the animal model, but also other pro-inflammatory cytokines such as IL-6. And at the same time, we again see significant upregulation of this anti-inflammatory cytokine IL-10, which we also saw in the animal model. We also see significant upregulation of cells expressing a specific marker called FOXP3. And these are T regulatory cells. And T regulatory cells are known to be very important in regulating the immune system and inhibiting the inflammation and keeping the immune system in balance. So we can see these immunomodulatory effects of reducing pro-inflammatory cells and cytokines and inducing anti-inflammatory cells and cytokine, both in the animal model and also in vitro in the test tube using cells from ulcerative colitis patients. We have then also studied biopsies taken from patients in our clinical studies. So here you go in and take a small piece of the colonic mucosa from the patients. And then you can take these biopsies and you can do section of the biopsies, and then you can stain for certain markers and then look in the microscope what type of cells you find in the colonic mucosa. And here we have taken biopsies at screening, so before they have received any dosing of Cobitolimod or placebo, and then 4 weeks after they have received 1 dose of Cobitolimod or placebo. And then we have looked at these IL-10 producing cells, so cells producing this anti-inflammatory cytokine IL-10. And we can then see a significant upregulation in the colonic mucosa of patients that have been treated with Cobitolimod, which you don't see in placebo-treated patients. Placebo-treated patient has the same level before and after treatment. We have also looked at cells expressing this FOXP3, which I mentioned on the previous slide, and that is then the T regulatory cells that are known to dampen the inflammation and regulate the immune system. And here again, we see significant upregulation in patients, in the colonic mucosa of patients that has been treated with Cobitolimod, which you don't see in placebo-treated patients. And then at last, we have also looked at this pro-inflammatory cytokine IL-17. And here we can again see a significant reduction in the colonic mucosa of patients that have been treated with Cobitolimod, which you don't see in placebo-treated patients. So these immunomodulatory effects that we see with Cobitolimod of inducing anti-inflammatory cytokines and reducing inflammatory cells and cytokine, we can have seen both in animal models, in vitro, and also in vivo using biopsies from patients in our clinical study after Cobitolimod treatment. So as a summary, in ulcerative colitis, you have an imbalanced immune system. You have too many of your pro-inflammatory cells and cytokines and too little of your T regulatory cells and anti-inflammatory cytokines. So you will create a chronic inflammation, which will lead to these ulcers and the symptoms that you see in ulcerative colitis. With Cobitolimod, we again receive a balanced immune system by reducing Th17 and IL-17, for example, so the pro-inflammatory cells and cytokines, and by increasing T regulatory cells and anti-inflammatory cytokines such as IL-10. So this balance that we then create in the immune system will then break this chronic inflammation, and you will have a well-functioning immune system again. And this will then lead to a healed colonic mucosa and improvement of the symptoms for the ulcerative colitis patients. This data, together with a lot of additional data that we have received, we have published in the scientific publication in the Journal of Crohn's and Colitis. So if you would like to dig more into this data and also additional data that we have on the mechanism of action of Cobitolimod, you can find all of that in this publication. So with that, I will end and hand over to Peter for a summary.
Thank you, Lotta. So just a summary then of the investment opportunity that you have in InDex. So we are addressing a disease that causes major suffering and social economic costs. With Cobitolimod, or we see that Cobitolimod has blockbuster potential, meaning sales of more than 1 billion per year, given the high unmet medical need for new treatment options and Cobitolimod's outstanding combination of efficacy and safety, and in combination with this novel mechanism of action. And we've come a long way. We're now in phase 3, the last stage of testing before you can apply for market approval. We've started the study, and we have financing secured until the next pivotal readout of clinical data. Thank you.
Thanks a lot, Peter. Thanks a lot, Charlotte. And for that, we open up for questions, but I could maybe start with the first question, because with this very strong data package, Peter and Charlotte, and also with the very low adverse effects for patients, now you scale up in humans. What problems will you usually face when you scale up? Because for me and for the audience as well, it looks very good. You did a very good homework already.
Yeah. The thing is, you never know what you find when you scale up. So, but we have no expected safety concerns. So we're not looking for anything specific. That goes for Cobitolimod, but we've also looked at the class of TLR9 agonists, and there's no class effect that we are concerned with. So we're quite confident that it will continue to show a very good safety profile. Then, of course, when you scale up, you don't, just scaling up in itself, you can find more rare events. But we have also increased the dose in the coming study. So it's the first time we tried 500 milligrams. The highest we tested before was 250. We have a big safety margin in animal studies where we tried much higher doses. But of course, you never know. But we absolutely do not expect that 500 would be any different than 250 or the lower doses. So I don't know if that's...
Yeah, well, that's a good answer, Peter. There's a question coming in here more related to the DIMS in a broader sense. If it addresses all immunological diseases. I don't know if it's Charlotte who answered that question, but
I can try to answer that.
Yeah.
As I mentioned earlier, depending on the sequence of the oligonucleotide, you can induce different immunomodulatory effects. So you can absolutely target other immunological diseases. So in this way, you can choose the oligo that best fits the immunomodulatory effects that you want to obtain for your specific disease. So in this way, it has a lot of opportunities to be used in different immunological diseases. And of course, also in other inflammatory diseases as well. I mean, we at InDex are investigating other DIMS compounds in different models for other inflammatory diseases. So there are absolutely potential to be used in other diseases than ulcerative colitis.
Thanks a lot. Thanks a lot. Do you expect, Charlotte, that with these good results and you get the balance back in the colon for the patient, that you are able to cure the patient totally, or are you still dependent on, as Peter mentioned in the beginning, you could look into the market for combining therapy? Are you still addicted to take other drugs after your treatment? Could you elaborate a little on this? I hope that the question is clear enough to answer.
I mean, we don't really know exactly how long the effect will stay for Cobitolimod. I mean, this study that we are doing now is the first time that we are investigating maintenance therapy. So we will, of course, have more answers to that after this. But I mean, as you mentioned, I mean, there is potential to use it in combination therapy as well, as it is a very good safety profile and also has a unique mechanism action. Of course, we need to also have more data on this for sure, but there is a great potential here.
Yeah, because the maintenance study you do in after you make the initial phase 3 study, you do the maintenance study afterwards, as I remember from earlier.
Patients that are responding to Cobitolimod in the induction study will then be re-randomized into maintenance study where they get Cobitolimod or placebo as maintenance treatment. So there you can more see the long-term effects of Cobitolimod, of course. Do you want to add something, Peter?
No. I mean, I don't think anybody's really claiming that they're out to cure the disease.
No.
Of the development programs that are out there, because it is a recurring disease. So we assume that we would have to continue to give this to prevent future flares. But
yeah, but if you
don't have the data.
Continue with your product with very low adverse effects, it will probably be very good for the patient.
Yeah, I mean, there is a good safety profile. It should be. We hope that it will fit very well for maintenance therapy as well, of course. Yeah.
Yeah. Then there's a couple of questions about DIMS in a broader sense. When will we get more info about the DIMS you're testing now? I know your focus is on Cobitolimod, but you have a nice portfolio. Could you answer that, Peter, or shall I?
Yeah. So as Lotta said, we are running testing some other DIMS in different models of inflammatory diseases. And unfortunately, those projects have been delayed due to COVID because we're working with labs where they were not allowed to go to the lab and they had to reduce the manpower. So those projects have unfortunately been delayed. But and Lotta maybe knows more, but we're still thinking that it will be this year that we will get results from those experiments that are aimed to confirm previous positive signals we've seen in earlier experiments. And some of those experiments are funded by the Swedish innovation agency Vinnova.
Have you mentioned earlier, Peter, what diseases you are looking into?
No, but I get the question often, and we just say other inflammatory diseases outside of ulcerative colitis, and that is because of patent purposes. Because if we now talk about what diseases those are, and everybody knows that there are different DIMS compounds we're testing, we could actually ruin our chances to get new patents for those. So that's why we have to be a bit secretive until we actually have the data, can file patents, and then can communicate the data more broadly.
Thanks a lot, Peter. Then a second question from the same person here about the PK study. I think you mentioned it briefly in the beginning, but how could you bridge the PK study to the phase 3 study?
Yeah, so actually we're not in a hurry with the PK study because we only need it for the filing of the drug, so it doesn't have to be done until we're done with the phase 3. But there are only a few patients, but then again, we're only in one clinic in Sweden. So it's a bit difficult to say when that will be done. It will depend a lot on the recruitment, but so far it's going well. They've found several patients already, but we don't really have a fixed timeline for that at this time.
No.
But that's an open study, so we know everybody there gets 500 milligrams of Cobitolimod, which is good in the sense that you know what they have and you can follow it real time. But on the other hand, from a scientific perspective, it's very difficult to draw any conclusions from it because you don't have a control group. So we don't know really what would have happened if we had a placebo group in there. So from an it's not designed to answer the question if Cobitolimod works or not. It's designed to find out where Cobitolimod ends up in the body.
Yeah.
Just as a reminder. And then the phase 3, we will continue to keep the market updated regularly on how we're doing in the study, and at the moment it's more getting the sites and countries up, and eventually it will be more focused on how many patients we actually have and when we think more exactly it can be done.
Yeah. There's a question about Cobitolimod as a pill, and I know we mentioned that on one of our earlier sessions. I think the question the topic last time, Peter, was Pfizer's acquisition of Arena, and they have a pill, and I think you also touched upon the area with pills, because that's a different mechanism.
It's actually the same mechanism. It's a different way of giving Cobitolimod.
Sorry, yeah.
So we have built a prototype of a pill or a capsule, which you eat it, so you take it through the mouth, but then it doesn't release Cobitolimod until it gets to the colon. So it's again colonic delivery, targeted delivery to the colon, but you eat it instead. So it's still a local treatment. So there we built a prototype, and now we are looking into, with these higher doses we're running in the phase 3 study, what the next step would be then for the oral formulation. So that's still under investigations. I don't have much to add there now today.
No, there's a little more. There's a question, as you see it as well there, regarding the DIMS, and if you could elaborate a little on what areas you target. But the question is more: is it rare diseases as orphan drug diseases, or is it more, major areas? But that's probably difficult for you to address.
Yeah. I think it's a bit of both.
Yeah. That's a good answer. Yeah. I don't know if we have further questions out there. No, doesn't really look like that. Good. With that said, I would like to thank Charlotte for a really good presentation, and also Peter, as usual, with a good overall view and good answers to the questions. And thanks a lot to the audience to participate in this event and put forward a good couple of questions for Peter and Charlotte. Thanks a lot, and enjoy your day.
Thank you very much.
Thank you.
Bye now.
Bye-bye.